Hello students, today we are going to start module 1 of our syllabus Physics 229. In this module, as a whole, you are going to understand blood and its properties. The very first topic that we are going to learn is basics of forensic serology. At the end of this lecture, you will be able to first, understand the basic terminology regarding forensic serology. Second, differentiate between plasma and serum. Third, understand the workflow for processing blood samples in forensic biology/serology laboratory. With respect to the key terminology, we will first start with serology. Serology methods are relatively simple and straightforward that deals solely with serum and its properties. And forensic serology. It is one of the subdivision of forensic biology that deals with the identification and examination of biological evidences. In particular, it focuses on determining the presence and identification of various bodily fluids such as blood, urine, saliva, and semen in a questioned sample. Bodily fluid stains are commonly associated with violent criminal cases. For instance, the identification of blood evidence is often necessary for investigating cases involving homicide, aggravated assault, drug intoxication, sexual assault, and burglary. Proving the presence of blood has probative value or may corroborate allegations of violent acts. The field generally is comprised of the detection of enzymes and antigens, as in the identification of seminal stains or blood typing, abo and secretor status, and DNA typing. Forensic hematology is the science that deals with the application of medical knowledge and detailed study of blood and blood components in order to aid in solving criminal and legal questions. Let's understand the difference between plasma and serum. The only thing one needs to remember is the use of anticoagulants during the collection time. If you want serum, the blood must be collected in plain vial, often denoted by red cap, and if you want plasma, the blood must be collected in vials with anticoagulants, denoted by different colors, yellow, purple, gray, etc. depending on the anticoagulant used. After the blood is collected, it will be centrifuged after specific time and you will get serum from the blood collected in plain vial. Serum is the liquid that separates from the blood when a clot is formed. On the other hand, the blood taken in the anticoagulant vial will give plasma. Plasma is the clear, straw-colored liquid portion of blood that remains after red blood cells, white blood cells, platelets and other cellular components are removed. Antigen, a substance, usually a protein that stimulates the body to produce antibodies against it. In forensics, abo, antigens present on the surface of RBCs, hence help in blood typing. Through blood typing, the forensic serologist ascertains the blood group of an individual which might be the source of the blood. Now, antibody is a protein that destroys or inactivates a specific antigen. Antibodies are found in the blood serum. Antiserum, blood serum in which there are specific antibodies. Abo blood grouping is done with the help of specific antibodies. For example, anti-A and anti-B. For inference, different types of control samples along with question samples are being collected from the crime scene and analyzed in the laboratory simultaneously. They are named as 1. Known, exemplar or reference, control. Are specimens from a known source, for example victim. Essential for comparison with DNA profiles from evidentiary specimens. 2. Alibi, alternative, known control. From a known source that may be the source of the evidence. 3. Blank control. A specimen known to be free of the item or substance being tested. To further avoid the contamination issues, the substratum comparison specimens are also taken. What does it mean? Substratum refers to the underlying material or surface on which the evidence is found. And A substratum comparison specimen is subjected to the same testing as the evidence. So, such specimen helps to detect interference in lab tests originating from the evidence surface. An unstained portion of the evidence underlying material is collected for this purpose. Now, let's understand the general workflow for processing blood samples in the forensic biology laboratory. When we get blood from the crime scene, the first step is to do visual examination. Then presumptive or preliminary tests are conducted to indicate the presence of blood. Afterwards, confirmatory or crystal tests are performed. The tests for species identification are done to determine whether it belongs to human or animal. The results of the species identification and confirmatory tests paves way for DNA profiling. In a nutshell, visual examination, presumptive tests, confirmatory tests and species identification are categorized in identification while DNA analysis is categorized under individualization. Now, all of you are able to interpret the basic terminology regarding forensic serology and also can identify the sequential workflow process adopted by the forensic laboratory for the analysis of blood evidence.